Cardiovascular physiology, cardiac electrophysiology. Welcome, listeners, to another episode where we delve into the wonders of human physiology. Today, our focus is on cardiac electrophysiology, the intricate study of the heart's electrical activity, which initiates and synchronizes every heartbeat. This electrical system is what makes the heart a truly self-regulating, or autorhythmic, organ. The principle of autorhythmicity, unlike skeletal muscle, which relies on external nerve impulses to contract, the heart possesses the remarkable ability to generate its own electrical signals. This unique property is known as autorhythmicity, or automaticity. Within the heart, specialized cells are responsible for creating and conducting electrical impulses, which ultimately trigger the contraction of the heart muscle, also known as the myocardium. The functional syncytium, unified contraction. The myocardium operates as a functional syncytium, meaning that individual cardiac muscle cells are interconnected electrically. This unity is made possible by intercalated discs, specialized junctions between cells that contain gap junctions. These gap junctions are tiny pores allowing ions to pass freely from one cell to another, creating a low-resistance pathway for electrical signals. Thanks to this configuration, action potentials can rapidly spread across the entire network of connected cells, resulting in a coordinated, wave-like contraction. The atria contract together as a unit, and the ventricles follow suit, ensuring efficient pumping of blood. The heart's conduction system, electrical pathways. The generation and propagation of electrical impulses in the heart follows a specific sequence, guided by a specialized conduction system. Let's walk through this pathway step by step. It all begins at the sinoatrial node, found in the right atrium near the entrance of the superior vena cava. This structure serves as the pacemaker of the heart, possessing the fastest rate of spontaneous depolarization and thus establishing the intrinsic heart rate, which typically ranges from 60 to 100 beats per minute. The sinoatrial node initiates the action potential, which then rapidly spreads throughout both atria. Next, specialized internodal pathways conduct the impulse from the sinoatrial node to the atrioventricular node, which is located in the interatrial septum near the tricuspid valve. The atrioventricular node acts as the heart's gatekeeper. It delays the electrical impulse for approximately 1/10 of a second, allowing the atria to complete their contraction and empty blood into the ventricles before the ventricles begin contracting. Additionally, it serves to filter excessively rapid atrial rates, protecting the ventricles from dangerously fast rhythms. The electrical impulse then travels through the bundle of His, which forms the only electrical connection between the atria and the ventricles, insulated by the heart's fibrous skeleton. From there, the signal moves down the right and left bundle branches along the interventricular septum, and finally to the Purkinje fibers. These fibers quickly distribute the impulse throughout the ventricular myocardium, ensuring a swift, coordinated contraction of the ventricles from the apex upward, which efficiently propels blood into the pulmonary artery and aorta. Cardiac action potentials, the basis of rhythm and contractility within the heart. There are two primary types of action potentials that are fundamental to its rhythm and contractile function. The first type is the pacemaker action potential, found in the sinoatrial and atrioventricular nodes. These cells do not maintain a stable resting potential. Instead, they spontaneously depolarize, which underlies the principle of autorhythmicity. The pacemaker action potential comprises several phases. The first is slow diastolic depolarization, where the membrane potential gradually rises until it reaches a threshold of approximately -40 millivolts. This process is driven by a slow inward current of sodium ions, a reduction in potassium ion efflux, and a brief influx of calcium ions through transient channels. Once the threshold is achieved, long-lasting calcium channels open, allowing calcium ions to enter and causing the cell to depolarize. Afterward, these channels close and potassium channels open, leading to repolarization and resetting the cycle. The rate at which the sinoatrial node generates impulses, and thus the heart rate, can be modulated by adjusting the slope of slow diastolic depolarization. Sympathetic stimulation, through hormones like norepinephrine and epinephrine, increases this slope, causing the threshold to be reached more quickly and raising the heart rate. Conversely, parasympathetic stimulation, via acetylcholine, reduces the slope and hyperpolarizes the cell, prolonging the time to reach threshold and decreasing the heart rate. The second type of action potential occurs in the contractile muscle cells of the atria and ventricles. These cells maintain a stable resting potential and feature a prolonged action potential that prevents sustained contraction. The phases include rapid depolarization driven by sodium influx, early repolarization with transient potassium efflux, and a distinctive plateau phase. During the plateau, there is a balance between slow calcium entry and potassium exit, resulting in extended depolarization. This ensures a lengthy refractory period, crucial for proper heart filling and preventing continuous contraction. The cycle ends with rapid repolarization as calcium channels close and potassium channels facilitate the cell's return to its resting state. The significance of refractory periods. The heart's refractory periods serve as vital safety mechanisms. During the effective refractory period, no new action potential can be generated, regardless of the stimulus's strength. This period coincides with the plateau phase and prevents overlapping contractions and sustained tension. Following this, the relative refractory period allows for the possibility of another action potential, but only if the stimulus is much stronger than normal. The electrocardiogram. A window into cardiac activity. The electrocardiogram, commonly known as the ECG, is a non-invasive recording of the heart's electrical activity, capturing the sum of all its action potentials. The P wave represents atrial depolarization, while the PR interval marks the time from the onset of atrial depolarization to the start of ventricular depolarization and reflects the delay at the atrioventricular node. The QRS complex signifies ventricular depolarization, during which atrial repolarization also occurs but is obscured by the large ventricular signal. The ST segment corresponds to the plateau phase when the ventricles are fully depolarized, and the T wave indicates ventricular repolarization. Lastly, the QT interval encompasses the total time required for both depolarization and repolarization of the ventricles. Bringing it all together, the integrated sequence. To summarize, the heart's rhythm unfolds in an elegant sequence. Cells in the sinoatrial node spontaneously depolarize, setting the pace. The impulse spreads across the atria, triggering their contraction and the P wave on the electrocardiogram. A brief delay at the atrioventricular node allows the atria to complete their function before the ventricles are activated. The impulse then rapidly travels through the conduction system, resulting in ventricular contraction and the QRS complex. Finally, the ventricles repolarize, represented by the T wave, and prepare for the next cycle. This sophisticated electrophysiological system ensures the heart beats rhythmically, efficiently coordinating its electrical activity with its role as the body's primary pump. Thank you for joining today's deep dive into cardiac electrophysiology, where science meets the art of life itself. Thanks for tuning in. If you enjoy the content, please like, share and subscribe. It's the Everything Surgery Podcast. Dissect the extraordinary.